Today, we are giving a presentation on the effects of cheese on the microbiota. So first off, cheese contains an incredibly diverse microbiota, with the bacterial community significantly playing a role in nutritional value and shelf life. High throughput screening, HTS, allows insight into many factors affecting the microbial community of cheese, allowing discovery of biotype diversity for discovery of dominant bacterial species and cheese samples. Seeing as cheese contains a very plentiful microbiota, our question is how that microbiota ends up affecting our own microbiome.
Lactic acid bacteria, also called lactobacillus, are gram-positive, rod-shaped or spherically shaped bacteria that can be found in decomposing plants and milk products. Their name comes from the fact that their metabolic product is lactic acid. Lactic acid in food can lead to spoilage after a period of time. These bacteria have increased tolerance to low pH levels, and this allows them to ferment. Labs that are used in probiotics fertilizer and the fermentation process, and it is found in a variety of foods. Because most species of labs cannot respirate, when used in a lab setting, the media it always includes a carbohydrate source.
Lactic acid bacteria, or LAB, often colonize fermented foods, including cheeses, during production and remain there during consumption. Increasing evidence seems to support the idea that the consumption of fermented foods can promote human health and affect the microbiota, acting as probiotics. Listeria monocytogenes is a pathogenic bacteria often found on several different foods, including dairy products like cheeses, and has the ability to survive being frozen and can also survive in environments with or without oxygen. Listeriosis is ranked third in total number of foodborne pathogen-related deaths in the U.S. 16 lab strains were tested for their ability to inhibit cell adhesion and invasion by L. monocytogenes. 6 strains were found to reduce adhesion, and 12 were found to inhibit invasion. In this same study, volunteers' fecal microbiota were analyzed before and after consumption of fermented milk containing Lactobacillus rhamnosus, an LAB strain. They were found to have a shift in the composition of their microbiota, specifically an increase in firmicutes and a decrease in Bacteroides species.
For the Bautista study, 16 different strains were used that were previously isolated during the ripening of an Italian hard cheese. These strains were collected and antibiotic resistance or susceptibility was tested. If the result was classified as being resistant to a breakpoint concentration, it was progressively tested with higher concentrations. This was done in triplicates. The pH balance was monitored daily throughout a period of 7 days using a pH meter. One of the strains was chosen to be used in the production of a probiotic fermented milk. Equal portions of this was administered to 14 healthy volunteers who followed an omnivore diet. They were divided into 2 groups, one group that consumed the milk and the second group consuming the placebo milk. Their fecal samples were collected at home and transported to the laboratory within 12 hours of collection. The RNA was extracted directly from the fecal samples using an electrophore Norgine RNA purification kit.
16 different strains previously isolated during the ripening of Italian hard cheese were used in this work. The strains were selected from a larger collection based on functional characterization that included testing for autoaggregation and hydrophobicity properties, as well as survival in simulated gastrointestinal passage. They belong to Lactobacillus 6 of them belonging to Lactobacillus helveticus, 9 to Lactobacillus rhamnosus, and 1 to Lactobacillus fermentum species.
The results of the study indicated a significant inter-individual difference in response to the intervention. This is possibly due to the relatively small sample size, and such inter-individual differences may not allow for a clear effect to emerge out of the data. There was no extremely clear effect on the major components of gut microbiota, similar among subjects when comparing intervention and baseline samples. However, they did find a shift in minor OTUs. OTUs are operational taxonomic units, and they're used to categorize bacteria based on sequence similarity. In particular, there was an overall increase in the firmicutes and a decrease in Bacteroides population as affected by fermented milk. Regarding the OTUs that changed as affected by the intervention, it was possible to observe a reduction in the relative abundance of Bacteroides in subjects during intervention compared to baseline, and an increase in the abundance of Prevotella. It has been recently reported that high Bacteroides levels are associated with an increased risk of weight gain and insulin resistance, while Prevotella is mostly associated with the production of short-chain fatty acids, or SCFAs. The protective roles of SCFAs against different types of disease is well documented. The protective roles range from maintenance of intestinal barrier integrity, mucus production, and protection against inflammation to the reduction of the risk of colorectal cancer. The main finding from the investigation of the intestinal microbiota of subjects that consumed fermented milk with or without a probiotic was that changes occur for minor bacterial populations. It has been proven through the Bautista-Gallego study that a ripened cheese harbors microbes that, when ingested in large quantities, have the potential to interfere and modulate the composition of the intestinal microbiota. It remains to be seen what the effect of these microbes is when ingested in lower concentrations or with closely related or competitive microbes in other fermented food. Even though there is a lot of evidence to suggest that cheese is helpful to your microbiome, we still need to see more studies with larger sample sizes to see more clear results.
As we know, gastrointestinal microbiota plays a significant role in more than just humans. A Korean study analyzed the effects of Bifidobacterium longum on healthy beagles by administering 10 grams per kilogram of body weight of queso blanco cheese. Cheese is a good source of protein, calcium, and vitamins for companion animals, but queso blanco has not been considered a supplement for pets until now. In this study, which was approved by the Institutional Animal Care and Use Committee of Chongbuk National University, 12 beagles were split into 3 experimental groups of 4 dogs each. One group received cheese containing B. longum, another received cheese devoid of B. longum, and the control group received no cheese at all. Fecal samples were collected at weeks 0, 4, and 8 of cheese intake. Following analysis of the fecal microbiota by 16S rRNA amplicon sequencing, it was found that Lactobacillus and Prevotellacea were present in the largest proportions. However, other families such as Fusobacteriaceae and Bifidobacteriaceae were also found. Fusobacterium, a pathogenic bacterium associated with bowel disease, was found to be reduced, and it can be assumed that B. longum inhibits the adhesion of Fusobacterium to the intestine and reduces intestinal bacteria count. Blautia, another bad bacteria, was found to have decreased in amount by 3 times in dogs compared to the control group. Ultimately, the study provided a variety of significant findings, but most importantly, it was revealed that queso blanco cheese containing B. longum is effective in improving fecal microbiota and even boosts the immune system. Beneficial intestinal bacteria were increased, while harmful bacteria were decreased. This study emphasizes how important the microbiota is in other species than humans, and shows the impact that the microbiology field can have elsewhere, such as in the veterinary sciences.
To conclude, it was previously stated that the lactic acid bacteria found within cheese may be able to act as a sort of probiotic and cause positive changes within our own microbiota. The Bautista-Gallego study supports this statement by demonstrating that ripened cheese in large quantities can be enough to intervene within our microbiota, thus altering its composition and promoting positive health changes. Unfortunately, one major pitfall of the study was the lack of a large enough sample size to clearly see the full effects that lab's microbiota has on our own. In the Park study involving the healthy beagles, each patient was given queso blanco, which contains Bifidobacterium longum. Over 8 weeks, various fecal samples were collected and analyzed, eventually showing a significant increase in the beneficial intestinal bacteria like Bifidobacterium, and a reduction in the amount of harmful bacteria like Enterobacteriaceae and Clostridium. Both of these presented studies served to reinforce the initial statement. Each of them incorporated stringent scientific methods to prove that the microbiota contained within cheese is enough to have a noticeable change on the microbiota composition of whatever may consume it through the introduction of new bacteria.
Thank you for listening, and if you have any questions, please let us know.